Okay, good day to all. Let us see 12th Zoology, 4th chapter, Principles of Inheritance and Variations, important 1 mark. If the mother is RH negative and fetus is RH positive, antibodies should be administered to the mother at 28th and 34th week of gestation as a prophylactic measure. The human Y chromosome is only 60 megabyte in size with 60 functional genes, whereas X chromosome are 165 megabyte in size with about 1000 genes. 23 pairs of human chromosomes include 22 pairs of autosomes, 44A, and 1 pair of sex chromosomes, XX or XY. The remaining 95% of the Y chromosome is referred as the non-combining region of the Y, NRY. Hemophilia disease was first reported by John Cato in 1803. The females are carriers of the disease and would transmit the disease of 50% of their sons even if the male parent is normal. Thalassemia is classified into alpha and beta based on which chain of hemoglobin molecule is affected. It is controlled by two closely linked genes HbA1 and HbA2 on chromosome 16. Phenylketonuria, it is caused due to mutation in the gene PAH, phenylalanine hydroxylase gene located on chromosome 12 for the hepatic enzyme phenylalanine hydroxylase. Huntington's chorea, the patients with this disease usually die between the age of 35 and 40. Each human diploid 2N body cell has 46 chromosomes, 23 pairs. Klinefelter syndrome, XXY males. This genetic disorder is due to the presence of one additional copy of the X chromosome, resulting in a karyotype of 47, XXY. Persons with this syndrome have 47 chromosomes, 44AA plus XXY. Turner syndrome, XO females. This genetic disorder is due to the loss of a X chromosome, resulting in a karyotype of 45X. Persons with this syndrome have 45 chromosomes, 44 autosomes, and 1 X chromosome. 44AA plus XO and are sterile females. In human beings, a normal female has 23 pairs of autosomes and a pair of sex chromosomes, 44A plus XX, and the male has 23 pairs of autosomes and a pair of sex chromosomes, 44A plus XY. Down syndrome, Klinefelter syndrome, Turner syndrome, and Part-2 syndrome are some of the chromosomal disorders. Down syndrome is due to trisomy of chromosome 21. Presence of trisomic condition of chromosome 13 results in Part-2 syndrome. Later, Oswald Avery, Colin McLeod, and Maclean McCarthy in 1944 repeated Griffith's experiment in an in vitro system in order to identify the nature of the transforming substance responsible for converting a non-virulent strain into virulent strain. When phage progeny was studied for radioactivity, it was found that it carried only 32 phosphorus and not 35 sulfur. The nitrogenous base is chemically linked to one molecule of sugar and the one carbon of the sugar. forming a nucleoside. When a phosphate group is attached to the 5 carbon of the same sugar, the nucleoside becomes a nucleotide. The nucleotides are joined, polymerized by condensation reaction to form a polynucleotide chain. The hydroxyl group and the 3 carbon of a sugar of one nucleotide forms an ester with the phosphate of another nucleotide. The chemical bonds that link the sugar components of adjacent nucleotides are called phosphodiester bond, 5'3' indicating the polarity of the strand. The ends of the DNA or RNA are distinct. The 2 ends are designated by the symbols 5' and 3'. The symbol 5' refers to carbon in the sugar to which a phosphate PO4 functional group is attached. The symbol 3' refers to carbon in the sugar to which hydroxyl OH functional group is attached. In RNA, every nucleotide residue has an additional OH group at 2 positions in the ribose. Understanding the 5'3' direction of a nucleic acid is critical for understanding the aspects of replication and transcription. The molecular biologists in the early 1980s, Leslie Urgel, Francis Crick, and Carl Woese independently proposed the RNA world as the first stage in the evolution of life, a stage when RNA catalyzed all molecules necessary for survival and replication. The term RNA world is first used by Walter Glippert in 1986, hypothesized RNA as the first genetic material on Earth. Andrew Freire and Greg Mello, recipients of Nobel Prize in 2006, were of the opinion that RNA is an active ingredient in the chemistry of life. In some viruses like tobacco mosaic virus, bacteriophage delta B, further today's OH group present at every nucleotide in RNA is area active group that makes RNA liable and easily degradable. The distance between 2 consecutive base pairs is 0.34 nanometer, 0.34 into 10 power minus 9 meter of the DNA double helix in a typical mammalian cell. When the total number of base pairs is multiplied with the distance between 2 consecutive base pairs, 6.6 into 10 power minus 9 into 0.34 into 10 power minus 9 meter base pairs, the length of DNA double helix is approximately 2.2 meter. The total length of the double helical DNA equal to total number of base pairs into distance between 2 consecutive base pairs. If the length of E. coli DNA is 1.36 mm, the number of base pairs in E. coli is 4 into 10 power 6, 4 into 106 micron, 1.36 into 1.3 meter divided by 0.34 into 10 power minus 9. The length of the DNA double helix is far greater than the dimension of a typical mammalian nucleus, approximately 10 power 6 meter. Kernberg proposed a model for the nucleosome in which 2 molecules of the 4 histone proteins H2A, H2B, H3 and H4 are organized to form a unit of 8 molecules called histone octamer. Neighboring nucleosomes are connected by linker DNA H1 that is exposed to enzymes. The DNA makes two complete turns around the histone ectomeres and the two turns are sealed off by an H1 molecule. Chromatin lacking H1 has a beads and a string appearance in which DNA enters and leaves the nucleosomes at random places. H1 of one nucleosome can interact with the H1 of the neighboring nucleosomes, resulting in the further folding of the fiber. The chromatin fiber in interface nuclei and mitotic chromosomes have a diameter that vary between 200 to 300 nanometer and represents inactive chromatin. 30 nanometer fiber arises from the folding of nucleosome chains into a solenoid structure having 6 nucleosomes per turn. DNA is solenoid and packed about 40 folds. The heavy culture was grown in a medium in which the nitrogen source NH4Cl contained the heavy isotope 15N and the light culture was grown in a medium in which the nitrogen source contained light isotope 14N for many generations. At the end of growth, they observed that the bacterial DNA in the heavy culture contained only 15N and in the light culture only 14N. The heavy DNA could be distinguished from light DNA 15N from 14N with a technique called cesium chloride CSCl density gradient centrifugation. The heavy culture 15N was then transferred into a medium that had only NH4Cl and took samples at various definite time intervals 20 minutes duration. After the second replication 40 minutes duration, they again extracted DNA samples and this time found the DNA settling into two bands, one at the light band position and one at intermediate position. E. coli that has 4.6 into 106 base pairs completes its replication process within 38 minutes in one strand, template strand with the polarity 3 days, 5 days. The replication is continuous and is known as the leading strand, while in the other strand, coding strand with the polarity 5 days, 3 days, replication is discontinuous, known as the lagging strand. The primer produces a 3 OH end and the sequence of ribonucleotides to which deoxyribonucleotides are added. The RNA primer is ultimately removed, leaving a gap in the newly synthesized DNA strand. It is removed from 5'N one by one by the exonuclease activity of DNA polymerase. The enzyme DNA polymerase can only catalyze the addition of a nucleotide to the new strands in the 5 days, 3 days direction, as it can only add nucleotides to the 3 days carbon position. The promoter is located towards the 5 days end. The terminator region located towards the 3 days end of the coding strand contains a DNA sequence that causes the RNA polymerase to stop transcribing. DNA-dependent RNA polymerase catalyzes the polymerization in only one direction. The strand that has the polarity 3 days, 5 days acts as a template and is called the template strand. The other strand which has the polarity 5 days, 3 days has a sequence same as DNA RNA except thymine instead of uracil and is displaced during transcription. This strand is called a coding strand. RNA polymerase 1 transcribes recombinant RNAs 28S, 18S and 5.8S, whereas RNA polymerase 3 is responsible for transcription of tRNA, 5sRNA and snRNA in capping and unusual nucleotide methylguanosine triphosphate is added at the 5 days end, whereas adenylate residues 200 to 300 poly A are added at the 3 days end in tailing. There are 64 possible triplets and 61 of them are used to represent amino acids. Since there are only 20 amino acids involved in protein synthesis, most of them are encoded by more than one triplet. Marshall-Nirenberg Shivara-Ohava enzyme polynucleotide phosphorylase called Ohava's enzyme, Hargobind Khorana, Francis Crick and many others have contributed significantly to decipher the genetic code. The genetic codon is a triplet code and 61 codons code for amino acids and 3 codons do not code for any amino acid and function as stop codon termination. 20 different amino acyl tRNA synthetases are known. The mRNA is translated starting at the 5 days end. The ribosome consists of structural RNAs and about 80 different proteins. The prokaryotic ribosome 70S consists of 2 subunits, the larger subunit 50S and smaller subunit 30S. The ribosomes of eukaryotes 80S are larger, consisting of 60S and 40S subunits. S denotes the sedimentation efficient, which is expressed as Swartberg unit. The 30S subunit of bacterial ribosome contains 16 srRNA and 50S subunit contains 5 srRNA molecules and 23 sRNA and 31 ribosomal proteins. The larger subunit in eukaryotes consists of 23 sRNA and 5 srRNA molecule and 31 ribosomal proteins. The smaller eukaryotic subunit consists of 18 srRNA component and about 33 proteins. UTRS are present at both 5 days end before start codon and at 3 days end after stop codon. The 5 days end of the mRNA of prokaryotes has a special sequence which precedes the initial AUG start codon of mRNA. The ribosome binding site is called the Sine-Dalgarno sequence or SD sequence. This sequences base pairs with the region of the 16 srRNA of the small ribosomal subunit, facilitating initiation. The subunits of the ribosomes 30S and 50S are usually dissociated from each other when not involved in translation. Initiation of translation in E. coli begins with the formation of an initiation complex consisting of the 30S subunits of the ribosome, a messenger RNA and the charged N-formyl methionine tRNA FMET-tRNA FMET. 3 proteinous initiation factors IF1, IF2, IF3, GTP guanine triphosphate and Mg2+. IF3 binds to the 30S and allows the 30S subunit to bind to mRNA. Another initiation protein IF2 then enhances the binding of charged formyl methionine tRNA to the small subunit in response to the AUG triplet. IF3 is released and allows the initiation complex to combine with the 50S ribosomal subunit to form the complete ribosome 70S. In this process, a molecule of GTP is hydrolyzed, providing the required energy, and the initiation factors IF2 and GDP are released. The next step in prokaryotic translation is to position the second tRNA at the A site of the ribosome to form hydrogen bonds between its anticodon and the second codon and the mRNA. Step 1. This step requires the correct transfer RNA, another GTP, and two proteins called elongation factors EFTS and EFTU. Once the charged tRNA molecule is positioned at the A site, the enzyme polypeptidase transferase catalyzes the formation of peptide bonds that link the two amino acids together. Step 2. At the same time, the covalent bond between the amino acid and tRNA occupying the P site is hydrolyzed, broken. The product of this reaction is dipeptide, which is attached to the 3' end of tRNA still residing in the A site. For elongation to be repeated, the tRNA attached to the P site, which is now uncharged, is released from the large subunit. The uncharged tRNA moves through the E site and the ribosome. The entire mRNA, tRNA, AA1, AA2 complex shifts in the direction of the P site by a distance of 3 nucleotides. Step 3. This step requires several elongation factors EFS and the energy derived from hydrolysis of GTP. This results in the third triplet of mRNA to accept another charged tRNA into the A site. Step 4. The sequence of elongation is repeated over and over. Step 5 and step 6. An additional amino acid is added to the growing polypeptide. Each time mRNA advances through the ribosome. Once a polypeptide chain is assembled, it emerges out from the base of the large subunit. The terminal codon signals the action of GTP dependent release factor, which cleaves the polypeptide chain from the terminal tRNA, releasing it from the translational complex. Step 1. The tRNA is then released from the ribosome, which then dissociates into its subunits. Step 2. In E. coli, nearly 260 genes are grouped into 75 different operons. The International Human Genome Project was launched in the year 1990. It was a mega project and took 13 years to complete. The human genome is about 25 times larger than the genome of any organism sequenced to date and is the first vertebrate genome to be completed. Human genome is said to have approximately 3 × 10⁹ base pairs. All the genes approximately 30,000 in human DNA. Although human genome contains 3 billion nucleotide bases, the DNA sequences that encode proteins make up only about 5% of the genome. On average, gene consists of 30,000 bases, the largest known human gene being dystrophin with 2.4 million bases. The function of 50% of the genome is derived from transposable elements such as line and ALU sequence. Genes are distributed over 24 chromosomes. Chromosome 19 has the highest gene density, chromosome 13 and Y chromosome have lowest gene densities. There may be 35,000 to 40,000 genes in the genome and almost 99.9 nucleotide bases are exactly the same in all people. Functions for over 50% of the discovered genes are unknown. Less than 2% of the genome codes for proteins. Chromosome 1 has 2,968 genes, whereas chromosome Y has 231 genes. Scientists have identified about 1.4 million locations where single base DNA differences, SNPs, single nucleotide polymorphisms, pronounced as SNPs, occur in humans. There are 23 pairs of human chromosomes with 1.5 million pairs of genes. Michelson and Stagel 1958 proved experimentally the semi-conservative nature of DNA replication using heavy isotope of nitrogen N15 in E. coli. In the 1958, Crick proposed that DNA determines the sequence of amino acids in a polypeptide protein through mRNA and proposed the central dogma of protein synthesis, which involves transcription and translation. Thank you.